University of California Cooperative Extension and University of California Davis Veterinary Medicine is bringing to you a recording from their March 2022 webinar series. This series was co-hosted by University of California Cooperative Extension Advisors Tracy Schohr, Grace Woodmansee, Rebecca Ozeran, and specialist Dr. Gaby Maier. Additional resources from this webinar and other CattleHealth videos can be found at ucanr.edu/sites/rangelands/CattleHealth. This session is focused on toxic plants and livestock, featuring Dr. Poppingay with the University of California Davis School of Veterinary Medicine. He works closely with the California Animal Health and Food Safety Laboratory to investigate toxic exposure to livestock and has a vast experience in diagnostic veterinary toxicology.
It's a real pleasure to be here. Tracy indicated that I'm with the California Animal Health and Food Safety Laboratory System. My lab is in Davis. Maybe many of you have submitted samples to us here in Davis. I'm the toxicology section head, and I work very closely with a number of analytical chemists that really make my job so much easier. Just a little bit more about my background. I've been in California for just around 17 years, back in 2004. Before that, I was on the East Coast at University of Pennsylvania for 10 years. And before that, in the Midwest, both Illinois and Michigan, the diagnostic labs there. So I think I have a really good sense of poisonous plants that vary based on the region. I expected really to see more cases in California when I moved out here. We certainly see a share of cases, but I've been a little surprised with the number of toxic plants that are in the environment that maybe we don't have more cases come through our laboratory system. So I'm just going to talk about poisonous plants. Let me just get this moving here. A couple of things that I'd like for you to be able to take away is some of the more common poisonous plants that we deal with in the diagnostic lab. Some of the species differences, because not all species are as sensitive to certain plants as other species. There's some significant variation, although some plants really do not discriminate in terms of their ability to intoxicate animals. They'll intoxicate any animal that ingests the plant in sufficient quantities. There's different ways you can categorize poisonous plants. I thought I would approach it the way we teach veterinary students, which is, what are the major organ systems affected? Is it primarily the heart? Is it primarily the nervous system, the GI, liver system, the renal system? In some cases, really no system other than acute neurologic impact that causes sudden death. A lot of the cases, the animals are just found dead. We can talk about some common exposure scenarios and factors that predispose to intoxication. And I think one of the key takeaways tonight would be to talk a little bit about how to investigate and resolve a suspected plant poisoning case, which would include appropriate diagnostics. I wouldn't expect you to buy any of these particular textbooks. I can show you the one I use here in the diagnostic lab. I don't know how well it's showing there. This is sort of the Bible of poisonous plants. The lead author was a veterinary toxicologist at Oklahoma State University. That's an in-depth resource. There's some that are a little bit more accessible. The Guide to Plant Poisoning of Animals in North America, that's a smaller book that has some decent pictures. The one thing I want to point out here, there is a tremendous resource in California that's specific to California, and that is a publication that is available free of charge. You can download the PDF. I put the link in there. The first website, it's a detailed publication it's entitled Livestock Poisoning Plants of California. And I think for a layperson's need, I think this is a great resource because it does cover most of the poisonous plants in California. It puts relative intoxications into context. There's a lot of great information, a lot of good pictures, and it's free of charge. Can't beat that. You can see there the top, that is the publication. A number of different co-authors. I think this was, yeah, January 2011. So there's not much that's changed over the years with poisonous plants. Maybe here and there, there's some new information, but I think this is still very relevant. The other great resource on the internet are a variety of different extension publications. So you can see there, I just put two on there from a quick search, University of Minnesota Extension, Plants, Poisonous Livestock, University of Missouri Extension, same title. There are a lot of extension publications out there, and I think they present really objective, up-to-date information with regard to poisonous plants. So that's a great resource. Obviously, our laboratory system, it's a situation where if you have a particular question, you can call the laboratory. They will generally connect you to me. I do have a veterinary resident that is in training who also is available to answer questions. So we have access to some resources that maybe would not be available elsewhere. And we can also go into detail with regard to how do you diagnose a problem, what kind of tests are available through the laboratory system to help confirm exposure. So a variety of different options with regard to what we can offer. One of the things that we do a lot of in calves is identify plants. And I think identification of plants is probably a great first step in trying to prevent an intoxication, knowing what plants are toxic and what plants are not toxic. We have a great resource on campus here. It's the Center for Plant Diversity, and we have some experts in the herbarium. So we'll receive samples of plants into the laboratory. They could be fresh or perhaps retrieved from a forage sample, and we will send it over to them for a positive ID. And they do a great job. If they can't identify a plant according to genus and down to the species level, they can generally get a genus. And I think sometimes it's just as important to rule out the non-toxic plant and possible exposure of animals to non-toxic ones to put the clients, kind of keep them a little less anxious that they're dealing with a toxic plant problem. Obviously, the Cooperative Extension is a great resource. Very often I'll refer people to the county extension agents, and very often they have experts that can go out and walk up a pasture and perhaps spot a potential problem. And I just wanted to bring this up to your attention. This is a USDA research lab out in Logan, Utah. It's called the Poisonous Plant Research Laboratory. They have experts there that research poisonous plants. Primarily, they focus on plants of interest in the western part of the US, but they also have quite a bit of information in terms of their publications. So if you wanted to dive a little bit deeper into some of these plants, there may be a research article that you could retrieve from their website. So in terms of general comments, poisonous plants definitely have an economic impact on livestock. This is some older information. I think it's really hard to come up with really precise economic impacts, but annual mortality for cattle and sheep is estimated to be 1% for cattle, 3.5% for sheep. So significant loss. I'm sure that in California, we miss a lot of intoxications, that just don't go, that are not diagnosed, they go undiagnosed. In terms of resources, that USDA publication, Plants Poisonous to Livestock in the Western States, that's another resource you could check into, and that may actually be free. You can see there that the authors make the statement that poisonous plants are a major cause of economic loss to livestock industry. Sort of similar type of percentages. The losses, direct losses from mortality, abortions, decreased production, birth defects, but there's also the additional economic impacts of trying to manage around poisonous plants through fencing, altered grazing, loss of forage. And I think it's important that with plant poisoning, we most often deal with acute illness. So animals suddenly become symptomatic, sudden death, but some plants are more of a chronic issue. They don't kill animals very quickly. They just slowly impact the animal so that the production decreases. They may eventually die, but it's over a long period of time. And we'll talk about some examples along those lines. But I'd say most of the poisonous plants we deal with, or at least that we recognize, are more acute intoxications. So what are some of the factors contributing to poisoning? So animal factors. Is it a species that's sensitive to a particular plant toxin? The best example there is with nitrates, probably one of the more common plant-associated intoxications that we see. Nitrates are a problem for ruminants, and that's because of the rumen. The rumen will degrade the nitrate to nitrite, and that's the nitrite that's absorbed and damages the hemoglobin. That reducing environment in the rumen isn't in a simple stomached animal, so we don't see nitrate intoxication, for example, in pigs. Really no cases in horses. Is the animal adapted to tolerate toxic plants? It is possible that exposure, low sublethal exposure over a period of time allows the rumen microbes to adapt. One example there is with oxalate containing plants. If the animal is allowed to gradually adapt, they can tolerate much higher concentrations in the plant than an unadapted animal. What other feed is available? The vast majority of toxic plants are not very palatable. There are a few exceptions, but that's the exception to the rule. If animals have access to other decent feed, they will avoid ingesting the toxic plant. In some cases, they can't avoid ingesting the toxic plant, and that's where a toxic plant may be incorporated into hay. So bales of hay, hay cubes. The animal is not able to select what they're going to eat, and that can be a risk factor. Introducing new animals into a new environment is always a risk as well. So animals get used to their environment, they figure out what maybe they can eat and not eat, and but sometimes in a new environment, they don't have that experience and can ingest something that's a problem. As far as the plant, we talked about palatability, the right time of the year. So you can have a toxic plant with toxins in the plant, but the concentration of the toxins in that plant may vary depending on the time of year you're talking about. Is it the early growth? Is it the later growth? Does the plant remain toxic when dried? And there's a lot of examples of both the fresh and the dried plant are equally toxic. So what kind of toxins are we talking about? One question is, why do these plants produce these things, these chemicals? Most of the time, it's thought that it's a defense mechanism on the part of the plant to protect the plant from predators, either insects or herbivores. A lot of cases we don't know. The diversity of chemical compounds in poisonous plants is tremendous. You can see some general categorizations there. I'd say we deal mostly with the alkaloids and the glycosides. So cardiac glycosides, cyanogenic glycosides, we deal with. We'll go through some of the alkaloid-containing plants a little bit as we go through here. The chemical structures vary quite a bit. And it's the actual chemical that our analytical chemists try to develop an assay for so that we could detect it in samples like the rumen or even antemortem in a serum or urine sample, maybe in the liver if the animal dies. So it's very important in some situations to be able to detect the specific toxin. So this is a tenet of toxicology. It's the dose that makes the poison. So it's not sufficient just to have a toxic plant in the environment and maybe an animal take a bite or two. The dose, the amount of toxin that the animal ingests may be insufficient to even cause clinical signs. So it may take a much larger ingestion to result in an adverse health effect. And plants do vary in terms of their toxicity. So I talk about nitrates. That's a relatively non-toxic chemical compared to something like conine, which is an alkaloid in poison hemlock. But it's also there in the plant at a much higher concentration oftentimes, and that's really where the high dose ingestion occurs. So the diagnosis of plant poisonings. This can be very difficult for a number of different reasons. A lot of times, if an animal's just suddenly found dead, there's no clinical signs. You don't know which major organ system might have been affected. There's maybe no evidence of ingesting a specific plant. Oftentimes, that's what we deal with when we have animals brought into us for autopsy. So very non-specific clinical signs. So if you have an animal that seizures, there's a number of different causes of seizuring or muscle tremors, not necessarily a plant-related problem. A lot of plants don't cause any lesions that a pathologist can actually detect. Especially the neurotoxic, the ones that affect the nervous system. When a pathologist looks under a microscope at the various organ systems, there's really no lesions at all. Identification of a toxin is often difficult. So we detect probably more of the toxins than a lot of other labs, but there's a lot of toxins that we don't detect. A good example there is with avocado. The toxin is believed to be a chemical called persin, but we can't develop a analytical test for it because we can't get the pure chemical that the analytical chemist can use to build the method. So in terms of approaching a case, pretty fundamental. If you're going to diagnose a plant intoxication, it's always great to find a toxic plant in the environment. So it's critical to have evidence of ingestion. And I've got some pictures here of some cases where really just looking at what the animals have been eating and identifying the plant can be the key to identifying a toxic plant problem. The clinical signs need to be compatible with the plant in question. So if it's a plant that causes liver damage, but the animal's dying from seizures, then it's not related to that plant that causes the liver damage. Rule out other causes of disease. There's lots of things that I mentioned that can cause seizures or muscle tremors or liver disease. And this is where really sometimes the first step is to do an autopsy, whether it's in the field or if you're close enough to one of our branches to actually bring the animal in. Your veterinarian can actually do a field necropsy and collect samples to submit to us that are going to help with the diagnosis. And we can advise you or the veterinarian as to what samples we'd like to get, what we can test, what we can't test for. When you're talking about purchased forages, I think it's incredibly important to get a representative sample. So if you have multiple bales of hay and you suspect a problem, we want to see more than just a quart bag full of sample, because poisonous plants can really vary in terms of their distribution in a batch of forage. So this is my lab crew up there at the top. This is our alkaloid screen that we offer. I think we probably are one of the few labs that actually detects specific alkaloids. And these alkaloids really just serve as a marker that if we find it, we know the animal was exposed to a particular plant. It doesn't say the animal was intoxicated, but it does at least confirm exposure. And we have some very sophisticated analytical platforms that allow us to detect these compounds in the low parts per billion range. So I couldn't do my job without our analytical chemists. And it's great to be able to have these kinds of screens, particularly if you have no other history to go along with a sudden illness or a sudden death. Recently, we did have some silage submitted to us from a producer that was concerned that the silage incorporated a whole bunch of poison hemlock. So we tested the silage for conine, which is an alkaloid in poison hemlock. Did not detect it, but it did put his mind at ease that there was no poison hemlock in the silage. So plant identification, fundamental step. Whether it's poisonous or not, it's useful to be able to know what plants are in the animal's environment. And I mentioned the herbarium that's been a tremendous resource for us. But if you're going to submit plants for identification, collect the entire plant, even with the root, and wrap the base of the plant in moist towels. And you can put that in a Ziploc bag, appropriate. It doesn't matter if you bend the plant, that's no big deal. But put it in a plastic bag, and it's best to probably then, if it's a fresh plant, to put it in a refrigerator. Different story with hay. There's no special way to handle hay. It's already dried out. Getting that representative sample is the critical part. One thing that we've sometimes asked, and it can be helpful, it's not maybe as good as having a real plant in front of you, but take pictures and send them to us via email. I've sent pictures over to the herbarium folks, and they come back at least to a genus, which may be sufficient. Sometimes contacting a local nursery can help you identify plants that you can't identify. I mentioned the extension agents that are very helpful. And I think it's important that, kind of getting back to the autopsy, making sure that we get the appropriate samples. So antemortem before the animal dies, things like blood and serum or urine are great. If the animal has died, GI contents oftentimes is the one specimen we really like to get. All of those samples are just saved fresh. There's no sort of, you can freeze them, they're going to be fine. The other type of samples that we like to get from a case go to the pathologist, and that requires putting tissue samples in formalin. That doesn't help us out as toxicologists, but it may be important for identification of very characteristic lesions. So looking at the GI content sometimes will pretty much make the diagnosis. Oftentimes, if we get a rumen content or a stomach content sample, particularly rumen content samples, we have a series of screens, and we'll take some of the rumen contents and start with the the largest aperture in the screens, and it goes down, becomes less and less. And in this particular case, this happened to be a goat that developed some heart problems going through the rumen contents. And we just put the rumen contents in there, flush it with water till we get rid of all the extraneous stuff. And lo and behold, in this case, you can see the fairly large leaf fragment that we found. And this actually turned out to be avocado. So that's an incredibly important step in terms of diagnosis. So this was one of the more interesting cases that I've been involved with here. This is a little bit unusual. This was not unusual back in Pennsylvania or the Midwest, but the history on this case, this was a goat herd of about 900 animals. This was a vegetation management goat herd, which I had never heard of such uses of goats until I came out to California. And these animals were being used on Mare Island, which is an old military base near Vallejo. At 6 a.m., about 500 goats were released into an area around an abandoned house for weed control. The people that were watching the goats knew that oleander was a problem, so they kept the goats away from oleander. By early evening, 32 goats were found dead. By the next day, 11 more goats were found dead. We actually went out and visited the site. And sudden death, we always sort of think of cyanide in plants or nitrate, nitrite. They were negative in the samples we tested. We did test a heavy metal screen, which was negative. Interestingly, with our alkaloid screen, we did detect taxus alkaloids. And the rumen examination identified taxus. So that's Japanese yew. And you can see the picture there in the lower right. The Japanese yew has very characteristic leaves. You're not going to miss them if you can find them. And I'll show you a picture in a minute. But fragments were taken from one or more of those goats. And you can see there, some laboratories are very good at looking at the microscopic appearance of plants and identifying the plant. This we sent to Texas Diagnostic Lab, and they actually look at sort of the superficial layer of the plant, the epithelial layer, and they look at the various pores in the plant, and that's very characteristic for certain plants. So this actually did turn out to be Japanese yew. And this is an aerial view of where these goats wound up being affected. So this is Mare Island, and that little circle there, the goats had been around where you can see the A, B, D, and E. Those areas were not believed to be involved in the illnesses of the goats, but you can see C there. And that actually was an old abandoned military housing. It was a big house. I assume it was a private residence at the time. And this was the building. So you can see the fence there, the temporary fencing. They put dozens of these goats in this area to try and take care of some of the brush. And this is what we found when we went and examined that area where the goats were. So it's a little bit hard on the left to say what that plant is, but on the right, if you look, these leaves are very characteristic of Japanese yew. And this gets back to the need to, when you can, to identify evidence of ingestion. So you can look at the plant, you can tell which plants have been nibbled on by animals, and that can be very helpful. So this was a case of Japanese yew. It is a very common ornamental in other parts of the country. I have not seen it quite as extensively used in California, but obviously this was a case where it was available to the goats. These are evergreen trees and shrubs. The leaves are very characteristic. They have the red berry there, which is the only part of the plant that is not toxic. And there's several different yew species. But looking at what is being fed to the animal can sometimes provide a clue. This was a case involving a group of horses that developed colic, and boy, their guts were just not moving. They were painful. We looked at some of the hay that was being fed, and all we found that was unusual were these little plant structures on the upper left. And as it turned out, that's a very characteristic plant structure for Jimson weed. And it turned out that these horses had ingested Jimson weed seeds. You can see there on the upper right, you can see the seed pod that's opened up, and there's lots of these little black seeds. And that's what the horses ingested. These plants contain atropine-like compounds that really just stop the gut. And it took a while for those horses to recover. But it was only that little fragment of plant material from the forage that gave us the clue. So that was Jimson weed, Datura stramonium. That's a very hardy plant. It can be found pretty much anywhere. The only good thing about Jimson weed is the animals would probably have to be almost starving before they would voluntarily eat it. Here was a case involving some performance horses. Sudden outbreak of GI signs in multiple horses at a horse park after introduction of a new grass hay, excessive salivation, gagging, again, colic. One of the things with horses that we deal with a lot is there's a suspicion anytime a horse gets colic that maybe it could be feed related, whereas the vast majority of times it's not anything bad in the feed. Horses just, if you look at them funny, can sometimes colic. But in these horses, sweating, rolling, pawing, decreased GI sounds similar to that other case, urgency to defecate, hives, which would be kind of an unusual allergic reaction. And we had forage submitted to us, and this was the plant that was identified. It's called death camus, which is a toxic plant. We didn't find any other alkaloids. This particular plant, we can't actually test for a specific chemical. This was one of those, well, maybe kind of cases. Maybe this plant in the hay could have caused a little bit of a problem in those horses. Certainly it was a significant finding. The death camus is more often associated with problems in sheep, pregnant sheep that develop congenital malformations, the cyclops lambs, although there is a neurotoxin in this particular plant. So this is from that extension publication, and this really gives you a good idea of what we see in the diagnostic lab. You know, I'm sure our cases are biased. You know, we do have assays for some of these toxins, plant toxins. We were one of the first labs to develop a chemical test for confirming exposure to oleander. So that probably results in more samples being submitted to us and more diagnoses. But this is getting to be a little dated now, but I don't think it's changed just based upon my experience. So over almost a 17-year period, oleander by far and away number one. Nitrate accumulators number two. Pyrrolizidine alkaloids, which I really hope we have some time to talk about because that's more of a chronic intoxication. Oxalate containing plants, oak, avocado. I mentioned the yew, yellow star thistle, which is a problem for horses, lupines, and then a non-toxic plant problem, and that's, we'll talk a little bit about the foxtails, which can be a real issue. In terms of dividing up the most frequent poisonous plant problems based on species, you can see there cattle, sheep, and horses, oleander, number one. In goats, interestingly, it's avocado. Number two for cattle, nitrate, nitrite, three oxalates, four pyrrolizidine alkaloids, five we put in there oak. You can see how this sort of goes across the different species. For horses, oleander, pyrrolizidine alkaloids, yellow star thistle, which is that centaurea, dogbane, and foxtail. And it's kind of interesting with regard to nitrate. Jump a little bit ahead here. Most of the cases or at least a lot of the cases are exposure through haze, and at least in cattle, it seems like the oat hay is particularly problematic. I would say the majority of cases in cattle where we actually get a sample in for testing of the forage, it turns out to be oat hay. So all organ systems can be affected. We're not going to have an opportunity to go through all of these. This is an incredibly useful table. I think that if you get that extension publication, it lists the various plants, the various toxins, but signs. What are some of the characteristic signs? And I think what really sort of stands out there to me is that a lot of these sudden death. So you may not even see any signs. The animal may be fine in the afternoon. You go out in the evening and the animal's dead. But there's a whole broad range of possibilities there. We occasionally do get some inquiries regarding ponderosa pine. We are working on an assay where we could actually take samples and detect the toxin. That's mostly associated with abortion. It really doesn't have any direct effect on the cow, and it's cattle by far and away. But anyway, that's a lot of information, but that's the type of information you'll see in that extension publication. So I'm going to talk a little bit about plants affecting the nervous system. A lot of these don't cause any characteristic lesions if the animal dies. You can see there's a long list. The ones I'll focus on primarily: the poison hemlock, Conium maculatum, the lupines, and nicotiana, the tree tobacco. We do have quite a lot of tree tobacco in California. Cyanide containing plants, pretty nasty. I'd say the cases that we see related to cyanide are fairly low. You can see there the milkweeds, the loco weeds, water hemlock, larkspur, bracken fern. These are all plants that in one way or another affect the nervous system. And we generally group these three plants together because the actual chemicals that cause the damage look a lot alike, and they actually work on the same receptors in the animal. So you can see the Conium maculatum, poison hemlock on the left, the tree tobacco in the middle, and then a lupine species there on the right. And I'm sure you've seen many of these plants. Not that this is a take-home message, but if you look at the different toxins that are in those three different plants, chemically they look very much alike. And in terms of adverse effects, one could say, yeah, they're going to affect the same receptor in the animal. And indeed, they do. They affect the nervous system and nervous transmission. I don't want to spend a whole lot of time on the various plant characteristics. You can look this up on the internet. Poison hemlock is a pretty easy one to ID just because of the characteristic of the plant, particularly the hollow stems with the purple spots is very characteristic. These are some pictures of poison hemlock. This is a big ubiquitous plant. I mean, this was very common in the Midwest and in Pennsylvania. Again, not very palatable. It's an early season growing weed, and it's that early growth you can see there in the middle that is particularly problematic. It can green up before a lot of other plants, and that may attract grazing animals to ingest it. The toxin in there, there's several different alkaloids that are very, very toxic, actually. And here you can see the characteristic purple splotching on the stem, and the stem is a hollow tube. There are some other plants that have hollow stems, but that's with the other characteristics. It's a fairly easy plant to identify. The tree tobacco, again, we do see it in certain parts of California. I don't recall seeing tree tobacco in the Midwest or the East, although occasionally just regular tobacco that's grown commercially can intoxicate animals. Some other plants that contain similar types of alkaloids. This is the tree tobacco. If you're ever in Davis by the vet school, we have a great example of tree tobacco right outside one of our buildings. And then the lupines, this is actually a pretty big genus, 200 to 300 different species of lupine. These are plants that you can buy in a garden store as well for sort of ornamental garden use around the house. If you've seen it once or twice, you probably know what a lupine looks like. And there's a lot of different flower colors, but they all have the basic botanical characteristics. You can see here, I think probably the one on the upper right is probably a garden variety of lupine. So you can see different alkaloids in the plant. Sometimes intoxication is not just related to one alkaloid, but multiple alkaloids. You can see there the toxicity, and I think this is important that all of those are toxic fresh if ingested, but they're also toxic if dried. So if they get incorporated into hay, that toxicity is retained. You can sort of see a toxic dose and the various species that are affected. So you can see poison hemlock, is going to be toxic for pretty much any species. You probably know that that's what Socrates used to kill himself. It was a extract of poison hemlock. Lab diagnostics for these particular plants, we can detect the alkaloids in various samples. And they're interesting plants because if you have a pregnant animal ingesting the plant during certain times of gestation, there are some congenital defects that can occur. And I'll show you a couple of pictures, but you can see there with poison hemlock cattle, if it doesn't kill the mother, it can affect the fetus. So especially between day 40 and 70 of gestation. Sheep, interestingly, are unaffected, but you can see for the other plants, there are similar windows of impacts on the fetus. So how do these plants result in congenital defects? Well, they keep the fetus from moving. So they immobilize the fetus, and this results in limb deformities, spinal cord deformities, cleft palate. And these are some examples. These are little pigs. You can see the deformities in the back leg there. The sow was exposed to poison hemlock, and the pigs were born alive, but they did have these deformities. And then this is a picture of calves that the mothers were exposed to lupine. You can see the cleft palate on the left, the little guy in the middle and the lower right is exhibiting spinal canal deformities, scoliosis, and that's the lupine. So changing gears here a little bit, this was kind of an interesting case, sudden death in goats. This was a backyard herd of 60 goats. Interesting diet. Diet consisted mostly of squash and cactus. I'm always amazed at what goats can eat. I guess that's why they're great brush clearing animals. They did have free access to a mineral block. This is not an unusual situation where a neighbor clipped what he believed to be olive trees in his yard and didn't want to have those clippings go to waste, so he donated the clippings to the goat herd. Within 4 hours of feeding the clippings, there were 3 dead goats, 7 very sick. They had a really fast heart rate. You could actually see the pulses in the neck, the jugular pulses. The animals weren't treated. A couple were sent in for some necropsy workup. Field necropsies were performed. Samples submitted to the diagnostic lab. Really not much on the field necropsy or autopsy. But we did detect cyanide, and it was detected in the liver, in the rumen contents, and they submitted some plant material that was fed, and you can see a picture of the berries and the plant, the lower right there. We tested that plant material for cyanide, and it was actually a very high concentration, and this was actually toyon. So toyon is an ornamental plant. We have some right around campus here. That's a cyanogenic glycoside containing plant. The genus species you can see there. You can see, I think this is a good example, is why we like to work with the scientific name, because the same plant can have a number of different common names. So you can see there the toyon can also be called Christmas holly, Christmas berry. So it is important to get that scientific name whenever possible. So there are a number of other cyanide containing plants. Sorghum is probably one of the more well-known, along with Prunus genus. So these are the cherries, but quite a few other plant species can contain cyanogenic constituents. And the cyanide actually in the plants is not all that toxic because it's a cyanogenic glycoside. So the cyanide is attached to sugar, and in that form, it's not much of a problem. But when that cyanogenic glycoside is broken down, that cyanide is released and absorbed and causes problems. So the problem with Johnson grass, it is used as a crop and as a forage. One of the problems is if you have the plant that freezes, the plant tissue is damaged, and that cyanogenic glycoside actually breaks down to release the cyanide. So when the animal ingests the plant, that cyanide is absorbed. And quite a few examples in the Prunus genus. You can see the western chokecherry there. These are trees, and so they're potentially a problem. So these are the cyanogenic glycosides. You can sort of see the cyanide, that C with the 3 bonds to the nitrogen, and the sugar that's detached, and that free cyanide causes intoxication. One of the real clues to differentiate cyanide intoxication from nitrate intoxication is the color of the blood. So if you're really astute and you look at the blood in a cyanide intoxicated animal, it is really, really bright red. It's cherry red. And that's because the cyanide attaches to some compounds that induce the blood to not release oxygen. So that's really super oxygenated blood. And that's what makes it really red. In the middle there, that's normal blood. And then on the right, that's blood taken from an animal with nitrate intoxication. And I mentioned that the nitrate is reduced to nitrite. It's absorbed. It affects hemoglobin. And the pigment then turns kind to a brownish hue to that blood. So that's a clue that a veterinarian or maybe even you could use in the field. or a pathologist here could use to at least raise the suspicion of either cyanide or nitrate intoxication. So cyanide is kind of one of those things, very, very potent, probably one of the more toxic toxins in plants. Animals that get a sufficient dose, they're going to die within probably 30 to 60 minutes. So very sudden death, labored breathing, frothing at the mouth, ataxia, muscle tremors, convulsions. I mentioned the bright red color of the blood. Really testing stomach contents or blood or plant material for cyanide is the way to go. It's one of those things that animals can ingest low levels, non-toxic amounts of cyanogenic glycoside-containing plants without any ill effect, because we actually in our body have a very good way to get rid of cyanide. It's only with the really large doses that you're going to run into a problem. All right, I'm going to put you to work here. This is maybe a difficult one, but can you identify this plant? Is this a loco weed, a yellow star thistle, pigweed, larkspur, or water hemlock? This actually is loco weed, which is another big category of poisonous plant. And the local weeds, there's actually two different genera, Astragalus and Oxytropis. It's kind of an interesting group of plants because it can cause several different problems. Probably one of the more dramatic is locoism. That's where the plant gets its common name. These are called vetches, milk vetches. There's a large number of species, and they contain a toxin called swainsanine. It's another alkaloid. And it's kind of an interesting compound because it prevents the animal from actually metabolizing carbohydrates. And when they can't metabolize carbohydrates, the carbohydrates build up into the cell in little vacuoles. And if it gets to a certain point, those cells are adversely affected. That's generally what we see with these two general plants. They also contain what are called nitro compounds. This is a neurotoxin that really affects the peripheral nervous system. Characteristic in cattle is what they call cracker heels or roaring disease. So it's a peripheral nervous problem. I'm not aware of it so much in California, but certainly in other parts of the West. These are plants that can accumulate tremendous amounts of selenium. In fact, they need high selenium in the soil to grow. And if livestock ingest the plants with the high selenium, over a period of time, they'll develop chronic selenosis. So some of the characteristics, I don't think it would take a herbarium specialist to be able to differentiate Astragalus from Oxytropis. I think you don't have to identify necessarily the species, although the species do vary in terms of which particular toxin they contain. And you can see there that at least with Astragalus bissulcatus, which is one of the more toxic ones you can see in the western part of the country, pretty wide distribution. That's the green part. I'm not sure why the Pacific coastal states are not included in that. But this is just a table to show you, depending on the species, they can either accumulate selenium, they can accumulate the swainsanine to cause the locoism, or nitro compounds that can cause the peripheral nervous problem. So clinical signs of abnormal neurologic behavior. I would say probably most cases reported in horses, but many other species can be affected. So depression, circling, incoordination, staggering gait, difficult to handle. One of the things is that up to a certain point, if you take them off the plant, they will recover. But once they reach a certain threshold, they do not recover. But even if they do recover, these animals are considered to be unpredictable. And if it's a performance horse, you don't want to continue using the horse in that manner. Infertility and reproductive problems, heart failure in cattle that are pastured at very high altitudes, just more of a chronic problem with weight loss and poor performance. These are the larkspurs. A few people mentioned the larkspurs. Another big genus of plants, a lot of different larkspurs in California, primarily a western state problem. Different species grow at different altitudes. Once you take a look at a couple of these, you can generally always at least identify a plant to that genus. You can see there larkspurs cause more fatal poisonings of cattle in the western US than any other native plant species. So these are native plants as opposed to introduced plants. Second only to local weeds in terms of economic losses. So you can see there some differences in terms of toxicity, cattle being the most susceptible, sheep actually much more resistant. Some variation in terms of toxicity based upon stage of growth, the season, duration of exposure. Interestingly, this is a plant, a toxic plant that is palatable. Animals will, particularly cattle, will ingest this plant voluntarily, often associated with sudden death. So here's another plant identification question. The following plant is one of the most toxic plants in California. It is an introduced plant, not a native, that is a common ornamental. It's the most frequent plant-associated poisoning of camelids and horses. So that should be a clue. And let me show you the plant. Here is the plant. Different flower colors, commonly planted along the interstate median strip. So is it a rhododendron, castor bean, oleander, eucalyptus, or a sago palm? So if Californians can identify at least one toxic plant, I think it would be the oleander, the Nerium oleander. Interestingly, the eucalyptus was mentioned by a couple folks. That is one that if you're just looking at the leaves can be confused with oleander. And I've got a picture to show you with regard to that. So the Nerium oleander does affect the cardiovascular system, as do all of these other plants. Some of these we do find in California. We've occasionally had some problems with rhododendron, and I mentioned the case with the yews. But by far and away, the Nerium oleander, they contain what are called cardioactive glycosides. So these are toxins that, like cyanide, are attached to a sugar, but they do break down in the body, and the toxin actually affects the ability of the heart to pump blood. It damages the heart. So this was a case in a llama, presenting complaint the animal was not eating, weight loss, sick for probably about a week, possible obstruction in the throat, came into the clinic. An obstruction was ruled out, but just wasn't eating, elevated heart rate, periods of abnormal heart rhythms, few gut sounds, lots of different things were done. And this animal actually had kidney failure, very high indexes of consistent with renal failure. And this was a case where, just based upon the clinical signs and the cardiac arrhythmias that were occurring, we did test for oleandrin, which is the toxin in oleander, and the urine and serum were positive for oleandrin. So we had a positive exposure and everything else fit. So this poor camelid actually had oleander intoxication. So here's the one plant that can be confused. You can see the oleander leaf there on the left, and then the eucalyptus leaf on the right. And so if we found portions of those leaves in a rumen contents, we'd have to differentiate those two. The oleander actually has a very characteristic pattern to the veins in the leaf as opposed to the eucalyptus. So it's not a hard one to identify. Here you can see the pattern, just that very prominent midrib and those very evenly spaced ribs that are going off to the side. So this is one of those plants that is toxic to pretty much any species. We've actually, kind of interesting, we've actually had a few human cases from coroner's office submitted to our lab for testing for oleandrin, because human labs don't test for oleandrin. And occasionally somebody knows that oleander is toxic and they want to get rid of somebody that they don't like, and will expose the individual to oleander. All parts of the plant are very toxic, the dried and fresh. One of the problems with oleander is that the animal doesn't have to have direct access to the plant. If the leaves dry up and then blow into an animal enclosure, and the animals ingest those dry leaves, that can wind up with significant adverse effects. Clinical signs you can see there within a few hours, although some of these animals will survive for a number of days. So it not only affects the heart, but a lot of these animals wind up with damaged kidneys as well. And this is what the pathologist would look at. So if a pathologist actually saw this in an animal, so you can see some of the hemorrhages in the heart, some more pale areas. This is the kidney. That is not normal. So that's the kidney damage there, and they can look under a microscope and characterize it further, and it's fairly characteristic for oleander. So does oleander and eucalyptus have the same color flower, is one of the questions. You know, I'm trying to think if I've ever actually seen a eucalyptus flower. The oleander, because it's an ornamental, actually has a number of different flower colors. So it can be red, it can be purple, it can be white. And maybe somebody else can mention something about eucalyptus. I can't. And so the diagnosis, identification of leaves, chemical analysis. You know, I want to make sure that we have plenty of time for a Q&A. And I know that someone has asked about acorns. Let me maybe go through this, and we will, I want to make sure I cover nitrates, and we can, you know, I'm thinking I actually took the acorns out, but we can talk about oaks during the Q&A. So this was another goat. This was a goat that vomited, projectile vomiting. And as you all know, ruminants generally don't vomit. They're not designed to vomit, really. And so whenever I get a call about a vomiting goat, or there's a vomiting goat in the history, I tend to think about one group of plants that we'll talk about. So this was a 1-year-old male goat, acute onset of vomiting, anorexia, abnormal behavior, probably in a lot of pain, wobbling, falling to the ground, salivation, teeth grinding, just really a gastrointestinal upset. Signs lasted for about 12 hours before it was presented to the clinic. And here was this poor little goat, vomiting rumen contents, crying out occasionally, just in a lot of pain. This was a goat that actually ingested rhododendron. And there are cardiotoxins in rhododendron called gray anotoxins. Some other related plants that contain the toxins, kalmia, the mountain laurel, the Japanese purus. You can see some others that are not as common. So gray anotoxins do affect the heart, but this is one category of toxicant that that causes that projectile vomiting in a ruminant. And I would say probably most of our cases involve goats. And you can see there, the upper left is the rhododendron, very common ornamental. The Japanese purus on the lower right, not as common in this part of the country. And again, you can see there the toxicity, 0.1% of body weight, clinical signs, kind of went over that. Diagnostic testing, we can test for gray anotoxins in urine, serum, GI content, and at least confirm exposure. Other cardiotoxic plants, I mentioned the Japanese yew. Avocado can be a problem, especially the leaves from the avocado. Most of our cases, again, have involved goats. Maybe we don't recognize it in other species. Number of plants affect the digestive system, and I'm just going to talk about the physical irritants, the foxtail barley, the bristle grass. There's some others that cause a pharmacologic effect on the GI tract to slow down peristalsis, but we actually do see quite a few grasses that can cause trauma, and this would be something to look for in hay. These are the sharp grass awns and barbed bristles that can cause injury to the oral mucosa. If they can get into the ear, they can go up the nose. Real problem in dogs. These things can migrate and cause abscesses. You can see there the clinical signs in livestock and horses. It can be a real challenge to try to remove these awns once they penetrate the mucosa or lodge in an ear or a nose. And you can see here some of the adverse effects that would indicate this physical irritation. And you can see the yellow foxtail. So I'm going to skip through photosensitization. So tansy ragwort is one that there's a question about. Been battling this weed on our ranch. How does it affect cattle if ingested? So the tansy ragwort actually, there's a number of different plants that contain the pyrrolizidine alkaloids. Those are the chemicals in the plant that cause the damage. This is not an acute plant intoxication. This is ingestion of a alkaloid containing plant over a period of time. The senecio, which the ragworts, that's the genus that the ragworts are in. The other common one in California is the fiddlehead. The Amsickia also contains pyrrolizidine alkaloids. So different genera contain the same type of chemical, and they're very common in along the west coast. As alluded to, it's hard to control. These are invaders of pastures and hayfields and open woodlands. I mean, these are really hardy plants, and we see it all over at this time of year as they start to emerge. This is one of the worst ones, the Senecio vulgaris, the common ground cell. Fairly characteristic leaves. They do have some fairly characteristic flowers. You can see that this is after the flowers have bloomed and gone to seed there. Kind of reminds you a little bit of a dandelion. And this is another one, the tansy ragwort. And this is the fiddleneck. And this is really the problem that we often see is when we get some hay in and we see these little white tufts of seed heads. Some of those are pretty benign, like the sow thistle, but if you find the common ground cell in a forage sample, that means the animal has been exposed. And I mentioned this is not an acute intoxication. So animals exposed to the pyrrolizidine alkaloids a little bit every day for several weeks. These pyrrolizidine alkaloids really target the liver, and it damages the liver cells, and those liver cells, in response, the liver develops a lot of fibrous tissue. So this really is a long-term problem with the liver. The liver eventually fails in these animals, but over that period of time, these animals can lose condition, lose weight, just not be very productive as that liver starts to degenerate. And the unfortunate thing is, in terms of diagnosis, is that oftentimes the exposure to the pyrrolizidine alkaloids occurred long before the animal actually becomes clinically ill. So you don't have representative forages or feeds available to test for the pyrrolizidine alkaloids. The pathologists, there are some characteristic changes in the liver that can suggest pyrrolizidine alkaloids is the problem, but we don't have a test for pyrrolizidine alkaloids. We can't take a liver sample and confirm exposure. We can test for some of them in the forage itself. And this was a case where actually alfalfa pellets had a PA containing plant incorporated in those pellets. There's no way you're going to identify the plant just looking at the pellet. But the animals that were fed these pellets are not going to have any choice but to ingest the pyrrolizidine alkaloids. Clinical signs, it's a chronic thing, liver disease. You can see there 2 to 8 months after first exposure. Neuroscience in horses are secondary to the liver damage. And it's just a really, it occurs every year. We have cases. It's frustrating to not be able to definitively diagnose it unless we find it in the feed. So ID in the hay, testing the feed, pathologic findings can be helpful, but there are other things that cause liver damage. Plain old oat straw can kill. What is the toxin in the plant that causes death? And most people selected nitrate, which is the correct answer. So I would say with nitrate, one of the common reasons is very high nitrate containing oat straw that's being fed to cattle. And, it just amazes me how high the levels can occur in some of these forages. There's a number of different plants that can accumulate nitrate. There's weeds, but there's also some crops like oat and corn. And really, it is a problem in drought-affected plants often, particularly with fertilized fields where a lot of nitrogen goes into the soil. The plant takes up the nitrogen. Normally, that plant would change that nitrogen into plant protein for growth, but the growth of the plant is stunted. So the nitrate is the nitrogen is converted into nitrate. It just sits in the plant, and when animals eat sufficient concentrations of nitrate, then they will become intoxicated. And it is, think back, it is ruminants. It's not simple stomach animals that are at increased risk. In this particular case, we found some pigweed, which is a notorious nitrate accumulator, and Sudan grass hay you can see there that 18,800 parts per million translates into 1.8% of that plant is nitrate. That's when you run into problems. So it can be related to heavy fertilization and the plant not being able to utilize the nitrogen. Highest concentrations are in the lower part of the plant. So sometimes you can cut the plant off at a higher level and not run into a problem. But if you fed the base of the plant, then you run into a risk of the nitrate intoxication. So clinical signs, generally these animals are found dead, similar to many of the other plants. But again, think about the fact that the blood could be very, very brown. And you can sort of see it here. You can see the affected blood that's on the left in that left picture. But if you just look at the animal, sometimes the muscle is very dark brown. Any blood that maybe is pooling there is just the pigmentation is off. And so diagnosis, we test for nitrate very often in ocular fluid. We can test in the feed. That's not a problem. So it's a very easy test to run. And maybe that's why we see more cases, because veterinarians know that we can test for it. So prevention, I'll let you consider some of the ways to prevent nitrate intoxication. And this is some information with regard to safe levels of nitrate. So actually plants can contain a fairly high concentration of nitrate and not cause a problem in a ruminant, but you can reach a threshold above which you can have sudden death. Okay, here's the oaks. Wide geographic distribution. Again, a lot of different species in that genus. They range in size from shrubs to tall trees. Actually, oaks can be used as useful browse for animals like goats. There's a lot of concern about acorns. That is a potential problem, but I think there's a greater risk probably of eating the leaves and especially the new buds at this time of year. The toxins in oak are called tannins. Tannins, anybody that drinks red wine and gets that characteristic tannin response, the same type of chemicals are in the oaks. These are very complex chemicals. They have an astringent effect on the gut mucosa, causing GI irritation. They react with constituents of the cells and denature protein and cause cell death. And they affect the kidneys. The GI tract often is affected, probably a little less the liver. And you can see there the tannins found in pretty much all parts of the plant. The young leaves and flower buds are especially toxic. Ripe acorns are less toxic than when green. And you can see their toxicity is not diminished by freezing or drying. I would not consider the oaks to be all that toxic. You can see their large amounts have to be ingested. This is the sort of the forage, greater than 10 kilograms per day per cow. A lot of anecdotal information that some animals just love to eat acorns. I know acorns are an important food source for wildlife such as deer. So I think the acorns, you'd have to ingest an awful lot of them to cause an adverse effect. So we can test for tannins in a urine sample, at least confirm exposure, but I would say it's not common to have a diagnosed case. And you can see there are some of the clinical signs related to the GI and the kidneys. Some animals actually can tolerate the tannins because they have proteins in their rumen content and their saliva that actually binds the tannins. So here are some pictures, some really nasty looking GI tracts. And then I'll end here with pine needle abortion. We occasionally get inquiries about pine needle abortion. When we do have beef cows that have aborted, it is something that our pathologists consider in a differential diagnosis. It's associated with ponderosa pine, the lodgepole pine, and the common juniper. There is a chemical in the plants called isocupressic acid that is metabolized, and it results in decreased blood flow to the uterus, increased release of fetal cortisol, and then a subsequent abortion. So the cow itself is not clinically affected unless there's a retained placenta or some problem associated with the abortion. You can see this chemical is found in both the bark and needles. The needles are of particular concern, and it does take a fair ingestion. So you can see maybe even up to 3 kilograms of pine needles per day for 3 days. And there is a window of susceptibility for that abortion is the last 3 months of pregnancy. So again, a large number of other things that probably need to be ruled out. You'd like to have a history of exposure. Hopefully at some point we would have an assay to be able to say the chemical is present in a biological sample. So these are some of the pictures of the trees. Interestingly, they often say that cattle will ingest if the cattle are kept underneath some of these trees and there's a storm coming through, and somehow that predisposes the animals to ingest the pine needles. I wouldn't think that would be a wonderful thing to have to eat, but they will eat the pine needles.
Just to add on that, one of the tips that we have suggested for producers in the area that I serve is being cognizant of where you're feeding supplemental hay, especially during the drought, and trying to avoid those areas to limit your risk and exposure.
Yeah, that's great. So one last comment there. So no effect from acorns on the fetus. That there is a condition called acorn calves that results in mostly a sort of a cephalic abnormality. In my 17 years here, I'm not aware of a case that's come in to our diagnostic lab. That doesn't mean it couldn't happen. But I don't think it'd probably be fairly uncommon.
We did get one more question in the chat. Should you be checking your hay before feeding it to prevent potential toxicity?
I always suggest that. As you feed it, it's probably good to open it up and just sort of, do a quick visual, survey of the feed. Sometimes, it's a lot of stuff to go through, and keep in mind that there isn't necessarily even distribution of a poisonous plant in a batch of forage. So you might have a hot spot. Nitrate is notorious for, of course, you're not going to see nitrate unless it's a weed like pigweed. If it's oat straw, you're not going to notice any difference. But yeah, I think it's a good idea. If you find something that doesn't look right, if you submit it to us, we can at least identify it maybe as a potential problem, or, a non-toxic plant. It's a little bit harder to identify plants in hay than it is if we get the whole plant, the whole fresh plant. But the herbarium folks are pretty doggone good at least getting to a genus.
Thank you to Dr. Poppingay for sharing your wealth of knowledge with livestock producers on poisonous plants during this session. Thank you for watching this webinar recording that was brought to you by University of California Cooperative Extension and University of California Davis Veterinary Medicine, and was co-hosted by University of California Cooperative Extension Advisors Tracy Schohr, Grace Woodmansee, Rebecca Ozeran, and specialist Dr. Gaby Maier.